Hi everyone, welcome back to my channel. Now we learn about Prokaryotic Capsule, Pili and Fimbriae. First coming to the capsule. Some prokaryotes have a layer of material lying outside the cell wall. This layer has different names depending on its characteristic. When the layer is well organized and not easily washed off, it is called a capsule. When it is a zone of diffuse, unrecognized material that is removed easily, that is called as slime layer. When the layer consists of a network of polysaccharides extending from the surface of the cell, it is called as glycocalyx, a term that can encompass both capsule and slime layers because they usually are composed of polysaccharides. However, some slime layers and capsules are constructed of their materials. For example, Bacillus anthracis has a proteinaceous capsule composed of poly-D-glutamic acid. Capsules are clearly visible in the light microscope when negative stains or special capsule stains are employed. They also can be studied with the electron microscope. Capsules are not required for growth and reproduction in laboratory cultures. They do confer several advantages when prokaryotes grow in their normal habitats. They help pathogenic bacteria resist phagocytosis by host phagocytes. Streptococcus pneumoniae provides a dramatic example. When it lacks a capsule, it is destroyed easily and does not cause disease, whereas the capsulated variant quickly kills mice. Capsules contain a great deal of water and can protect against desiccation. They exclude viruses and most hydrophobic toxic materials such as detergent. Many prokaryotes have a regularly structured layer called an S layer on their surface. In bacteria, the S layer is external to the cell wall. In archaea, the S layer may be the only wall structure outside the plasma membrane. The S layer has a pattern something like float tiles and is composed of protein or glycoprotein. In gram-negative bacteria, the S layer adheres directly to the outer membrane. It is associated with the peptidoglycan surface in gram-positive bacteria. It may protect the cell against ion and pH fluctuations, osmotic stress, enzymes, or the predaceous bacterium Dello vibrio. The S layer also helps maintain the shape and envelope rigidity of some cells. It can promote cell adhesion to surfaces. Finally, the S layer seems to protect some bacterial pathogens against host defenses, thus contributing to their virulence. Coming to the Pili and Fimbriae, many prokaryotes have short, fine hair-like appendages that are thinner than flagella. These are usually called fimbriae. A cell may be covered with up to 1000 fimbriae, but they are only visible in an electron microscope due to their small size. They are slender tubes composed of helically arranged protein subunits and are about 3 to 10 nanometer in diameter and up to several micrometers long. At least some types of fimbriae attach bacteria to solid surfaces such as rocks in streams and host tissues. Fimbriae are responsible for more than attachment. Type 4 fimbriae are present at one or both poles of bacterial cells. They can aid in attachment to objects and also are required for the twitching motility that occur in some bacteria such as Pseudomonas aeruginosa, Neisseria gonorrhea, and some strains of Escherichia coli. Type 4 fimbriae are also involved in gliding motility by myxobacteria. These bacteria are also of interest because they have complex life cycle that include the formation of a fruiting body. Many bacteria have about 1 to 10 sex pili per cell. These are hair-like structures that differ from fimbriae in the following ways. Pili often are larger than fimbriae. They are genetically determined by conjugative plasmids and are required for conjugation. Some bacterial viruses attach specifically to receptors on sex pili at the start of their reproductive cycle. Coming to the flagella and motility, most motile prokaryotes move by use of flagella, that is, thread-like locomotor appendages extended outward from the plasma membrane and cell wall. Bacterial flagella are the best studied and they are the focus of this discussion. Bacterial flagella are slender, rigid structures about 20 nanometer across and up to 15 to 20 micrometer long. Flagella are so thin they cannot be observed directly with a bright field microscope, but with stained with special techniques designed to increase their thickness. The detailed structure of a flagellum can only be seen in the electron microscope. Bacterial species often differ distinctively in their patterns of flagella distribution, and these patterns are useful in identifying bacteria. Monotrichous bacteria. Mono means single and trichous means hair. Have one flagellum. If it is located at an end, it is said to be polar flagellum. Amphitrichous bacteria. Amphi means on both sides. Have a single flagellum at each pole. In contrast, lophotrichous bacteria. Lopho means stuffed. Have a cluster of flagella at one or both ends. Flagella are spread fairly evenly over the whole surface of peritrichous bacteria. Peri means around. Coming to the last one, that is flagellar ultrastructure. Transmission electron microscope studies have shown that the bacterial flagellum is composed of three parts. The longest and most obvious portion is the flagellar filament, which extends from the cell surface to the tip. The second one, a basal body, is embedded in the cell. The third one is a short curved segment, that is the flagellar hook, links the filament to its basal body and act as a flexible coupling. The filament is a hollow, rigid cylindrical constructed of subunits of the protein flagellin, which ranges in molecular weight from 30,000 to 60,000 daltons, depending on the bacterial species. The filament ends with a capping protein. Some bacteria have sheaths surrounding their flagella. For example, Vibrio has a membranous structure surrounding the filament. Vibrio cholerae has a lipopolysaccharide sheath. The hook and basal body are quite different from the filament, slightly wider than the filament. The hook is made of different protein subunits. The basal body is the most complex part of a flagellum. In Escherichia coli and most gram-negative bacteria, the basal body has four rings connected to a central rod. The outer L and P rings associated with the lipopolysaccharide and peptidoglycan layers respectively. The inner M ring contacts the plasma membrane. Gram-positive bacteria have only two basal body rings, an inner ring connected to the plasma membrane and an outer one probably attached to the peptidoglycan.